In this presentation, we're going to learn about proteins. Proteins are known as multi-purpose molecules. They are the most structurally and functionally diverse group. The functions are involved in just about anything in a living organism. Um, Proteins function as enzymes. Enzymes are molecules that help facilitate chemical reactions, specifically speed up the chemical reactions in the body. Structure, so proteins make up the structure of living organisms. So for example, keratin, which makes up the structure of your hair, collagen, which is a protein that causes your skin to be elastic. Proteins function as carriers and transport. They actually move things throughout the body. For example, hemoglobin is a protein that helps transport oxygen through your blood. Aquaporins are proteins that help water move through cell membranes. They also help with cell communication. Believe it or not, cells talk to one another. And so cells talk to each other by sending proteins, which are called protein signals. Insulin and other hormones are considered signaling molecules. They can also function as receptors. So you have proteins that receive these signals that allow the cells to talk to each other. Proteins are used in defense, specifically talking about immune defense or your immune system. Proteins called antibodies are what allows your body to build a memory of what a pathogen or something that would make you sick coming into your body. Movement, so your muscles are made of two proteins, actin and myosin, and together those proteins are responsible for all your muscle movement. And proteins are used for storage, not necessarily in animals, but definitely in plants, specifically legumes, beans. So the energy is stored within the seed as protein. So the structure of proteins, the monomer of a protein is an amino acid. There are 20 different amino acids that can be found. The polymer of a protein is something called a polypeptide. And the reason we call them polypeptide is the bond that holds amino acids together is called a peptide. And because proteins are made of lots of amino acids, there's lots of these peptide bonds. So we call it a polypeptide, many peptide bonds. So here we can see one amino acid, so one monomer of protein being joined with another monomer of a protein, another amino acid, and this is going to be done by dehydration synthesis. Remove the OH from this amino acid and the H from the other to join them together into a polymer. Here are some examples of proteins. Proteins become functional when they are folded into a three-dimensional shape, and it's the three-dimensional shape of the protein that determines its function. Amino acids. The structure of amino acids is that they have in common, all amino acids have a central carbon, an amino group, a carboxyl group, and then what's different about each amino acid is the R group, also known as a side chain. And this is the variable group. Just like we use X in an algebra equation, we use R to represent variable groups on a molecule. And so these R groups are different for each amino acid, and it's what gives the identity to each of the 20 amino acids. So here's our central carbon. Over here is our amine. This is the carboxyl group, and this part here is known as the backbone of the amino acid. This is the same between every single amino acid you're going to find. The amine group, the central carbon, and the carboxyl group. This is also why we call them an amino acid. This is the amino part, and this is the acid part of our amino acid. What's different is the R group. So coming off of the central carbon, we can add all sorts of different side chains, which gives the identity to our 20 different amino acids. Some of the R groups are nonpolar and hydrophobic, and the way that you can tell that these are nonpolar and hydrophobic is that the R groups are made of just C's and H's. With the exception of methionine, we have a sulfur. However, the sulfur is not protruding from the chain. It's actually central in the chain, so it does not act as a polar group. Same with the nitrogen here in tryptophan. We have polar amino acids, so ones that are polar or charged, that are hydrophilic, that will easily mix with water, and the way that we tell this is because they have functional groups sticking off of it. So here in serine, we see a hydroxide. Here in threonine, we see a hydroxide sticking off. A sulfhydryl on a cysteine, so on and so forth. So these are polar and hydrophilic because of the functional groups that are sticking off of the R group. So the different effects of the R groups. So amino acids with hydrophilic R groups are going to turn outward to mix with water in the cell. The amino acids with hydrophobic R groups are going to turn inward to hide from the water. So here's our polypeptide. Each one of these circles represents an amino acid. The ones that are red are representing amino acids with hydrophobic side chains or R groups, and the blue are ones that are hydrophilic. And so what happens is when we add this polypeptide to water, it causes the polypeptide to fold and form our protein shape. Here we have the hydrophobic amino acids hiding on the inside and the hydrophilic pointing outward. We also have amino acids that ionize, meaning that they are either acidic. So here, these happen to be the acidic side chains. They donate an H+. We also have some that are basic. That means that they're able to accept an H+. And then we have two amino acids that have sulfurs in them, and what they form are what are called disulfide bridges. Disulfide, so di meaning two, sulfide referring to the sulfur, and bridge referring to a covalent bond. So an R group with a sulfur will bond with other R groups with sulfurs to form covalent bonds and give a strong three-dimensional structure to our protein. So here's a disulfide bridge, the sulfur of one R group bonding covalently to the sulfur of another R group. And this will give a nice three-dimensional shape to our protein. So building proteins. We build proteins by building peptide bonds. And a peptide bond is simply the bond between the carbon of one amino acid and the nitrogen of another. And the way that we get this bond between the carbon of one and the nitrogen of the other is by removing an H and OH, which is called dehydration synthesis. And the result, the bond here between the C and the N, so the carbon of the first amino acid and the nitrogen of the other, is called a peptide bond. Peptide bonds are covalent, so they're nice and strong. Polypeptide chains have direction to them. We have the N-terminus, which means the nitrogen end, and we have the the carbon terminus, or the C-terminus, the carbon end. The function of the protein depends entirely on its structure, its 3D twisted shape. So the way that we twist and fold that polypeptide, that chain of amino acids, will determine the shape of our protein and therefore the function of the protein. And we have some proteins that are globular, like hemoglobin. That's why the globin means globular or round and three-dimensional. Or we have some that are filamentous, long strands like collagen. There are levels of organization to a protein, and the first level of organization, also known as the primary structure of a protein, is the order of the amino acids. The amino acid sequence is determined by our DNA, and a slight change to the amino acid order can completely change the shape of the protein that is made and therefore the function. So here's our polypeptide made of lots of amino acids. So here we have a strand of DNA which gives us our instructions for building our polypeptide. The polypeptide then folds to form our three-dimensional protein. We get the right shape. However, if we have a mutation to our DNA, so down here it says mutant DNA even though it's cut off, so if there's a change to the DNA, we call it a mutation, and mutations to the DNA cause the amino acids to be in a different order. So here we had phobic, 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 phobic amino acid, then philic. Here we have phobic, phobic, philic, phobic, philic. And by doing this, it's going to change the way our polypeptide folds, and therefore it's going to change the way that our protein behaves. Wrong shape. So the wrong order of amino acids give you the wrong shape, and therefore the protein can no longer do its job. An example of this is sickle cell anemia. If we take a look here, we have the sixth amino acid in the chain, glycine, is changed into a valine. And This amino acid here, glycine, is hydrophilic, and valine is hydrophobic. This one little change out of 146 amino acids causes our red blood cells to be a nice donut shape, which is its normal shape, will change to be the sickle shape. Secondary structure is known as local folding. This is folding along short sections of the polypeptide, and this is influenced entirely by the backbone of the amino acids, not the R group. So hydrogen bonds will form between the carboxyl group of the backbone of one amino acid and the amine group of the backbone of another. And so it's actually interactions between the backbones that either cause our proteins to fold into what are known as alpha helix, which is the spiral shape, or into what's known as a beta pleated sheet. If you've ever folded a piece of paper into a fan, that's the shape of a beta pleated sheet. So again, another depiction of our alpha helix, the coil, or the beta pleated sheet, the folded paper. Tertiary structure, so tertiary means third level of organization. This is where the R groups really come into play. This is whole molecule folding. This is where the protein will get its three-dimensional shape. And it gets its three-dimensional shape from hydrophobic interactions, meaning the hydrophobic R groups hiding on the inside of the protein. And we also get hydrogen bonding and ionic bonding occurring. So we get, between our hydrophobic groups, we can get some hydrogen bonding going on. We can get what are called ionic bonds, or between our acid and base R groups forming these ionic bonds. And then we can also get covalent bonds forming between the sulfurs of R groups, also known as our disulfide bridge. And all of these things, so these hydrogen bonding, the ionic bonds, the disulfide bridges, give our protein a three-dimensional shape. And it anchors this three-dimensional shape. At the tertiary form of the protein, this is when the protein becomes functional. Then the fourth level is what's called quaternary structure. And what happens with this is more than one folded polypeptide chain bonds to another. So here with collagen, we actually have 1, 2, 3 polypeptides that have been folded into their tertiary form, interacting to form a quaternary. So quaternary structure is multiple tertiary forms of proteins interacting together. Hemoglobin, we have 1, 2, 3, 4 folded polypeptide chains. And they are attracted by their hydrophobic interactions. Okay. So again, a big overview. The first level of organization is the primary structure, which is quite simply the order of the amino acids. The secondary structure is going to be local folding. Interactions between the backbones of amino acids. AA means amino acids. Tertiary is going to be the hydrophobic interactions, disulfide bridges, hydrogen bonds, ionic bonds between the R groups. And then the final level of organization is going to be the quaternary form, where we have multiple polypeptides interacting together. So denaturation is the process by which a protein unfolds or loses its shape. By unfolding, the protein will no longer be able to do its job. Denaturation can be caused by changes in temperature, changes in pH, changes in salinity, meaning how much salt is available, and it alters the secondary and tertiary structures of the protein, causing the protein to lose its three-dimensional shape. And if you lose the three-dimensional shape, you lose the function of that protein. And that's all I have to say about proteins.